What if some of the most important guardians in your life are so small you will never see them, and so quiet you will never feel them working? Right now, inside your body, millions of tiny molecules are drifting through your blood and the clear currents of your lymph. They are shaped like delicate Y-shaped keys, each one designed to recognize a single unfamiliar shape in the vast molecular world around you. These are antibodies, silent protectors created by your immune system, constantly searching for the things that do not belong. Tonight, we're going to move gently through the quiet science of these remarkable molecules, how they are made, how they recognize invaders, and how they help keep you healthy every day without you ever needing to notice. Just listen to each fact as it arrives, slowly and calmly, and let the story of the smallest guardians in your body carry you softly towards sleep. Somewhere inside you, right now, there are tiny molecules shaped like the letter Y, drifting through your bloodstream with a quiet sense of purpose. These are antibodies, proteins made by your immune system, each one a small and exquisite sculpture of intention. They are not alive, not in the way you or I are alive, but they carry within their shape a kind of knowing, a memory written in folds of amino acids that allows them to recognize things that do not belong. They float through the rivers of your blood and the still pools of your lymph, patient and unhurried, like lanterns set adrift on a dark and gentle sea. And though they are impossibly small, so small that billions of them could rest on the tip of your finger without you ever feeling their weight, they are among the most important structures your body has ever learned to build. They are your oldest companions, and they have been with you longer than almost anything else. The word scientists use for antibodies is immunoglobulins, a name that sounds heavy and clinical but means something rather beautiful when you break it apart. Immuno, from the Latin immunis, meaning free or exempt, a word that once described those who were spared from service or obligation, and globulin, meaning a small round protein, something globe-shaped and quietly complete. So an immunoglobulin is, in the gentlest translation, a small round thing that sets you free. It is a name that speaks not of war or battle, but of release, of being let go from the grip of illness, of being carried safely past danger by something too small to see. Scientists often shorten the word to IG, a soft abbreviation you will hear again tonight, tucked before letters like G and A and M, each one naming a different kind of guardian in this vast and tender army that lives inside you, working without rest, asking nothing in return. Antibodies are born from a kind of white blood cell called a B cell, and the story of their creation is one of the quietest miracles in all of biology. B cells themselves are born in your bone marrow, that soft, hidden tissue nestled deep inside your bones, the place where your blood is made. It is warm there and dark and impossibly busy, like a city that never sleeps but never makes a sound. Each B cell that emerges from the marrow carries on its surface a unique version of an antibody, a prototype, a draft of something that might one day be needed. Most of these B cells will drift through your body and never find their match, never be called into service. But some, the ones whose shape happens to fit an invader the body has never seen before, will awaken and begin to multiply and fill your blood with copies of themselves and the antibodies they carry. It is a story of patience and of readiness and of a kind of hope written into your very cells. Every antibody is designed to recognize one specific target, and that target is called an antigen. The word antigen comes from antibody generator. It is the thing that calls the antibody into being, the shape that gives the antibody its reason for existing. An antigen might be a fragment of a virus's coat, or a protein on the surface of a bacterium, or a molecule carried by a grain of pollen drifting through the spring air. It is not the whole invader that the antibody recognizes, but rather a small specific patch on its surface, a molecular fingerprint, a signature written in the language of shape and charge. And the antibody reads that signature the way your fingers might read the face of someone you love in the dark, not by seeing, but by touching, by fitting, by knowing through the intimacy of contact. This lock and key relationship between antibody and antigen is one of the most precise and elegant partnerships in all of nature, a recognition so specific it borders on devotion. If you were to look at an antibody under a powerful enough microscope, you would see that its Y shape is not merely decorative. It is functional, meaningful, like the shape of a hand or the curve of an ear. The two upper tips of the Y are where the magic happens. These are the variable regions, and they are the parts that reach out and bind to an antigen, gripping it with a specificity so fine that even a single atom out of place would break the connection. Each antibody's tips are unique, sculpted by the particular arrangement of amino acids that make up their surface. Meanwhile, the stem of the Y, the lower portion, is more constant, more shared among antibodies of the same class, and it serves as a signal to the rest of the immune system, a kind of flag that says, "I have found something, come and help." And so the Y shape is both a sensor and a messenger, a molecule that reaches out into the unknown with one end and calls home with the other. Here is something that might take your breath away if you let it settle in slowly enough. Your body can generate an almost incomprehensible diversity of antibody shapes. Modern estimates suggest the potential combinations number in the quintillions, far beyond what was once thought. That is not a number the mind can hold. It is larger than the number of stars in the observable universe, larger than the grains of sand on every beach the Earth has ever known. And yet your body carries this potential quietly, without effort, without fanfare, written into the shuffling and recombining of a handful of gene segments that rearrange themselves in every developing B cell. It is as if your immune system were a poet with an infinite vocabulary, capable of writing a unique verse for every threat it might ever encounter, even threats that do not yet exist. This staggering diversity is one of the great wonders of biology, a quiet testament to the creativity written into the deepest code of your being, humming along beneath your awareness as you rest. Antibodies do not stay in one place. They are travelers, wanderers, drifting freely through the rivers and tributaries of your body's fluids. You will find them in your blood, of course, that warm, ceaseless current that carries oxygen and nutrients to every corner of your being. But you will also find them in the clear fluid of your lymph, in the thin film of moisture that coats your lungs, in the quiet reservoirs of fluid that bathe your tissues. They move without muscles, without legs, without wings, carried instead by the gentle currents of your circulation, the slow tides of fluid that ebb and flow through spaces between your cells. They are everywhere and nowhere, present in concentrations so vast that a single drop of your blood contains millions of them, each one a tiny sentinel floating in the warmth of you. And though they cannot think, cannot feel, they are always oriented toward the same purpose, to find what does not belong and to hold it still until help arrives. Your immune system has two great branches, two ways of protecting you, and antibodies belong to the one called the adaptive immune system. The other branch, the innate immune system, is older, simpler, and faster. It responds to threats the same way every time, without learning, without remembering, like a wall that stands against every storm equally. But the adaptive immune system is something different, something more personal. It learns, it remembers, it gets better with time, and antibodies are its most eloquent expression, molecules that are tailored, shaped, refined to recognize specific threats with a precision that the innate system could never achieve. The word adaptive is a gentle one, and it means exactly what it sounds like. This part of your immunity adapts to the world around you, changes with you, grows as you grow. It is the part of your protection that carries your personal history, the molecular diary of every germ you have ever met, every cold you have ever weathered, every vaccine you have ever received. There is a tenderness in the way antibodies work because they do not kill. That may surprise you. We so often hear the language of warfare when we speak of the immune system, of battles and attacks and defenses. But antibodies themselves are not weapons. They are markers, identifiers, gentle hands that reach out and hold on to something dangerous so that other, more powerful parts of the immune system can come and deal with it. When an antibody finds its matching antigen, it binds to it and holds on, and in doing so it changes the surface of the invader, coating it in a signal that immune cells can read. It is less like a soldier and more like someone tying a ribbon around something that needs attention, a quiet, persistent act of pointing and saying, "Here, this one, this is the one that does not belong," And in that pointing, in that patient holding, the antibody has done its most essential work. The rest of the immune system takes it from there. Antibodies can also do something remarkably gentle and effective. They can neutralize toxins simply by binding to them. When a bacterium releases a harmful substance into your body, or when a venomous creature delivers its poison, the toxin molecules have a specific shape that allows them to interact with your cells and cause damage. But if an antibody finds that toxin and wraps itself around the active part of the molecule, it blocks the toxin the way a hand might cover a keyhole, not by destroying the key, but by simply preventing it from fitting into the lock. The toxin is still there, still floating in your bloodstream, but it can no longer do what it was designed to do. It has been silenced, muffled, rendered harmless by the quiet embrace of a molecule that asked for nothing in return. This is neutralization, and it is one of the most elegant acts of protection in all of biology. A defense that works not through force, but through the simple, unhurried act of holding on. Among all the molecules your body makes, and it makes thousands upon thousands of different kinds, antibodies are among the most specific. Their ability to distinguish one antigen from another, to tell the difference between two molecules that might differ by only a single atom, is almost beyond comprehension. This specificity is not an accident. It is the result of hundreds of millions of years of evolution, a long and patient refinement that has produced molecules capable of reading the molecular world with a sensitivity that no human technology has ever fully matched. Laboratories around the world use antibodies as tools precisely because of this extraordinary precision, to detect diseases, to identify cells, to measure the tiniest concentrations of substances in the blood. And all of this specificity lives inside you right now, carried by molecules so small they are invisible, so numerous they are uncountable, so faithful they have never once forgotten what they were made to do. You are, in the most literal sense, a library of recognition, and every antibody is a book that knows its reader by heart. Not all antibodies are the same. They come in 5 distinct classes, 5 families, each with its own personality and purpose, and learning their names is like learning the names of 5 quiet guardians who have been watching over you since the day you were born. Their names are IgG, IgA, IgM, IgD, and IgE, the letters standing for immunoglobulin, followed by a single letter that marks each class like an initial on a handkerchief. Each class has a different shape in the stem of its Y, a different weight, a different way of moving through your body, and a different role to play in the long, patient work of keeping you safe. Some are abundant, flooding your blood in vast numbers. Others are rare, held in reserve for moments of particular need. Together, they form a complete and overlapping system of protection, a constellation of 5 quiet lights, each one shining in its own way, each one illuminating a different corner of your defense. Of the 5 classes, IgG is the one you carry the most of in your blood. It is the most abundant antibody in your circulation, making up roughly 70 to 75% of all the antibodies flowing through your veins right now. If your bloodstream were a river, IgG would be the current itself, quiet, steady, everywhere at once. It is the antibody of persistence, of long-term protection, the one that remains in your system long after an infection has cleared or a vaccine has been given. IgG is the memory keeper, the one that ensures your body remembers a threat for months, sometimes years, sometimes an entire lifetime. It moves easily out of your blood and into your tissues, slipping through the walls of your smallest blood vessels to patrol the spaces between your cells. It is both common and extraordinary, the workhorse of your immune system, the antibody that does the most work with the least fanfare, humming along in the background of your life like a lullaby you have heard so many times you have forgotten it is playing. IgG holds a distinction that no other antibody class can claim. It is the only one that can cross the placenta, that extraordinary organ that connects a mother to her unborn child. During pregnancy, IgG molecules from the mother's blood pass through the layers of the placenta and enter the bloodstream of the developing baby, carrying with them the mother's immune memory, her history of encounters with the world. This means that a newborn arrives into the brightness and noise of life already wrapped in a borrowed immunity, already carrying antibodies against diseases the baby has never encountered but the mother has. It is a gift given without words, without awareness, a molecular inheritance passed from one body to another through the most intimate connection two human beings can share. This transferred immunity fades over the first few months of life as the baby begins to build its own. But for those early vulnerable weeks, the mother's IgG is there, a shield woven from love and biology, indistinguishable from each other. IgA is the antibody of surfaces, of boundaries, of the places where your body meets the outside world. You will find it in your saliva, coating the inside of your mouth with a thin and invisible layer of protection every time you swallow. You will find it in your tears, those quiet messengers of emotion that also happen to be carrying molecular guardians in every drop. You will find it in the mucus that lines your airways, in the fluids that bathe your digestive tract, and, perhaps most beautifully, in breast milk, where it is passed from mother to nursing child in every feeding. IgA is the antibody that stands at the gates, the one that meets the world before the world can get inside. It works by binding to pathogens on your mucosal surfaces and preventing them from attaching to your cells, a kind of gentle refusal, a turning away at the door. It does not cause inflammation, does not raise alarms. It simply and quietly says, "Not here, not today," and the invader drifts away without ever knowing how close it came. Here is a quiet surprise. Even though IgG is the most abundant antibody in your blood, IgA is the most produced antibody in your entire body when you count everything. All the surfaces, all the secretions, all the hidden linings where it is made and released. Your body produces several grams of IgA every single day, a staggering output for a molecule so small, a testament to how much of your protection depends not on what happens deep inside your blood, but on what happens at your borders, your surfaces, the thin and delicate membranes where the inside of you meets the vastness of the outside world. Most of this IgA is secretory IgA, a special form that is designed to survive the harsh environments of your mouth, your gut, your lungs, places full of enzymes and acids that would destroy other proteins in moments. Secretory IgA is built for endurance, for standing watch in difficult places, and it does so with a patience that mirrors the patience of the body itself. When your body encounters something new, something it has never seen before, the first antibody it sends into the fray is IgM. IgM is the antibody of first response, the earliest voice to speak when danger arrives, the first draft of a defense that will be refined and improved in the days and weeks that follow. It appears in the blood within days of an infection, rising quickly while the more precise and longer-lasting IgG is still being prepared. Because of this, doctors often look for IgM in the blood as a sign of a recent or active infection. Its presence is a whisper that says something new has arrived and the body is learning to deal with it. IgM is not as specific as IgG, not as finely tuned, but it is fast and it is generous, binding to threats with a broad and open embrace that buys the immune system time. It is the first chapter of every immune story your body writes, the opening line that makes all the rest possible. IgM has a shape unlike any other antibody class, and it is breathtaking in its geometry. While most antibodies travel alone as single Y-shaped molecules, IgM joins together in groups of 5, arranging itself into a structure called a pentamer. 5 Y shapes linked together in a ring, like 5 pairs of hands joined in a circle. This pentameric form gives IgM 10 binding sites instead of the 2 that a single antibody would have, which means it can grip onto an invader with an avidity that compensates for its lower individual precision. It is strength in numbers, strength in connection, a molecule that has learned that sometimes the best way to hold on to something is to reach out with many hands at once. The pentamer is also large, much larger than a single IgG, which means IgM tends to stay in the bloodstream rather than slipping into tissues. It is a guardian of the blood itself, a sentinel that patrols the central rivers of your circulation with a quiet, 5-pointed vigilance. IgE is the rarest antibody in your blood under normal circumstances, present in concentrations so low they are measured in nanograms, billionths of a gram. And yet, for all its scarcity, IgE plays an outsized role in two of the most dramatic responses your immune system can mount: allergic reactions and the defense against parasites. When IgE encounters an allergen, pollen, dust, a fragment of peanut protein, it binds to the surface of cells called mast cells and basophils. And when that allergen appears again, the IgE molecules trigger those cells to release a flood of chemicals, including histamine, that cause the swelling, itching, and sneezing of an allergic response. It is a system built for a different world, a world where parasitic worms were a constant threat, and the inflammatory response IgE triggers was a necessary defense. In the modern world, where parasites are rarer but allergens are everywhere, IgE sometimes overreacts, mistaking the harmless for the dangerous. It is a guardian whose vigilance in certain moments becomes its flaw. In the warm stillness of your bloodstream, IgE is the quietest presence of all. Under normal conditions, it is the least abundant antibody circulating through your body, present in amounts so vanishingly small that for many years scientists struggled to detect it at all. It was not identified as a distinct antibody class until 1966, decades after the other classes had been named and characterized. Its discovery was the work of two separate research teams, Kimishige and Teruko Ishizaka in the United States, and SGO Johansson and Hans Benisch in Sweden, who arrived at the same conclusion through different paths. The rarity of IgE in the blood is not a sign of unimportance, but of specialization. Most IgE is not floating freely. It is bound to the surfaces of mast cells in your tissues, waiting there like a sentinel perched on a wall, ready to respond the moment the right antigen arrives. Its scarcity in the blood is simply because it has already found its post, already taken its position, already settled into the quiet work of watching. IgD is the most mysterious of the 5 antibody classes, the one that scientists understand the least, the one that still holds its secrets close. It is found mainly on the surface of immature B cells, those young immune cells that have recently left the bone marrow and are still learning their role, still waiting to encounter the antigen that will give their life direction. On the surface of these B cells, IgD sits alongside IgM, both of them acting as receptors, as antennae, as the B cell's way of sensing the molecular world around it. When a matching antigen brushes against these surface antibodies, the B cell wakes up, activates, and begins the process of multiplying and producing antibodies in earnest. But beyond this role as a surface receptor, the purpose of IgD remains largely unclear. Small amounts of it are found in the blood, and some research suggests it may play a role in activating certain other immune cells, but the full story of IgD is still being written, still unfolding in laboratories around the world. There is something almost poetic about the fact that, after more than a century of studying antibodies, one of the 5 classes still resists complete understanding. IgD remains an open question, a gentle mystery at the heart of immunology. Scientists have proposed many possible roles for it: that it helps regulate the immune response, that it plays a part in defending the upper airways, that it may signal other immune cells in ways we have not yet fully mapped. Some studies have suggested that IgD may be involved in the body's response to certain bacterial infections, particularly in the tonsils and adenoids, those quiet clusters of immune tissue at the back of your throat. But no single definitive function has emerged with the clarity that we see for IgG or IgA. And perhaps there is a lesson in that even even within the body's most studied systems, there are still rooms we have not entered, still stories that have not been told. The mystery of IgD is a reminder that the universe inside you is as vast and unexplored as the one above you. What makes each antibody class different from the others is the shape of the stem of its Y, the constant region, the part that does not change from one antibody to the next within the same class. This stem is made of a particular type of heavy chain protein, and there are 5 types of heavy chains, one for each class: gamma for IgG, alpha for IgA, mu for IgM, delta for IgD, and epsilon for IgE. The heavy chain determines everything about how the antibody behaves beyond its ability to bind an antigen, how long it lasts in the blood, whether it can cross the placenta, whether it triggers inflammation, which immune cells it communicates with. It is like a letter written in the same handwriting but on different paper, sent through different channels, arriving at different destinations. The tips of the Y may be infinitely variable, shaped by the randomness of gene recombination, but the stem is a family trait, a class identity, a quiet declaration of purpose that tells the rest of the immune system exactly what kind of help has arrived. The story of how antibodies come into being begins in one of the most hidden and sheltered places in your body, the bone marrow. This soft, spongy tissue fills the hollow centers of your largest bones, your hip bones and your vertebrae, and the long bones of your arms and legs. And it is there, in that warm and quiet darkness, that B cells are born. The marrow is a place of extraordinary activity, producing billions of new blood cells every single day. And among those billions are the B cells that will one day become the source of your antibodies. Each B cell begins as a stem cell, a blank page, and through a carefully orchestrated series of genetic rearrangements, it develops its own unique antibody, one that has never existed before and may never exist again. The bone marrow is the nursery, the cradle, the first chapter of every antibody's story, and it does its work in a silence so deep that you will never hear it, no matter how closely you listen. Every B cell that leaves the bone marrow carries on its surface a unique antibody, a molecular signature that belongs to that cell alone. Think of it as a fingerprint, but even more personal, because while your fingerprints are shared across all the cells of your skin, each B cell's surface antibody is entirely its own, different from the antibody on every other B cell in your body. This surface antibody serves as the B cell's receptor, its way of sensing the world, its means of detecting whether a matching antigen is nearby. The B cell drifts through your blood and lymph, presenting this unique receptor to everything it encounters, asking a silent question over and over again: are you the one I was made for? For most B cells, the answer will always be no. They will circulate for weeks or months and eventually die without ever finding their match. But for the rare few whose antibody happens to fit an invader, the answer will be yes, and everything will change. The moment a B cell encounters the antigen that matches its surface antibody is one of the most significant events in your immune life. It is a recognition, a meeting, a molecular handshake between two shapes that were made for each other. When the antigen binds to the B cell's receptor, a cascade of signals is triggered inside the cell, chemical messages that travel from the surface to the nucleus, telling the B cell that its time has come, that the threat it was designed to recognize has arrived. This activation usually requires a second signal from a helper T cell, another type of immune cell that has independently verified the threat and now gives the B cell permission to respond. It is a system of checks and balances, a safeguard against false alarms, and it ensures that the immune response is targeted and deliberate. When both signals are received, the B cell awakens from its quiet wandering and begins to transform, preparing for a period of furious multiplication that will fill the blood with antibodies. Once activated, a B cell begins to divide rapidly, making copy after copy of itself in a process called clonal expansion. The word clonal comes from clone. Each new cell is an identical copy of the original, carrying the same unique antibody, the same precise fit for the antigen that triggered the response. Within days, a single activated B cell can give rise to thousands of daughter cells, all of them bearing the same molecular specificity, all of them pointed at the same target. It is an act of extraordinary amplification, like a single musician being multiplied into an entire orchestra, all playing the same note, all directed towards the same purpose. This expansion happens primarily in the lymph nodes, those small bean-shaped structures tucked throughout your body, where B cells gather and divide in a fever of activity that you might feel as a slight swelling or tenderness in your neck or under your arms. That tenderness, that gentle ache, is the sound of your immune system composing its response, note by urgent note. As the clone of activated B cells expands, some of the daughter cells undergo a remarkable transformation. They become plasma cells, large, specialized factories whose sole purpose is to produce and release antibodies into the body's fluids. A plasma cell is a B cell that has given up everything else. The ability to divide, the ability to recognize new antigens, the ability to do anything other than its one appointed task, in order to dedicate itself entirely to the production of antibodies. Under a microscope, plasma cells are distinctive. They are larger than most immune cells, with a vast internal network of protein-making machinery called the endoplasmic reticulum, which fills nearly the entire cell like the coils of a great and tireless engine. Everything about the plasma cell is oriented toward output, toward production, toward the single-minded release of the molecule it was born to make. It is a cell that has found its purpose and surrendered to it completely, and there is something deeply moving in that devotion. A single plasma cell can release roughly 2,000 antibody molecules every second. Let that number settle into you slowly, like a warm breath on a cold night. 2,000 molecules, each one perfectly shaped, each one carrying the same precise specificity, launched into the bloodstream every single second. Over the course of a plasma cell's life, which may last anywhere from a few days to several weeks, it will produce and release millions upon millions of antibodies, an output so vast and so relentless that the cell eventually exhausts itself and dies. It is a brief and brilliant life, a life spent entirely in service, and the antibodies it leaves behind continue to circulate long after the cell itself is gone. They drift through your blood and lymph, finding their targets, marking invaders, neutralizing toxins, doing the work that the plasma cell gave everything to make possible. There is a kind of quiet heroism in that, a cell that lives fast and gives everything, and whose legacy floats on in the warm rivers of your body. Not all of the B cells that arise from clonal expansion become plasma cells. Some take a different path. They become memory B cells, and their role is not to fight the current battle, but to remember it. Memory B cells are long-lived, patient, unhurried. While plasma cells burn bright and fast, memory B cells settle into the quiet corners of your immune system, your lymph nodes, your spleen, your bone marrow, and wait. They can survive for decades, carrying within them the molecular memory of an antigen they encountered perhaps only once, years, or even a lifetime ago. And if that antigen ever returns, if the same pathogen ever tries to enter your body again, the memory B cells will recognize it instantly, awaken, and mount a response that is faster, stronger, and more precise than the first. This is the foundation of immunological memory, the reason a disease that once made you very sick may barely affect you the second time. Your memory B cells are the keepers of your immune history, and they never forget. The existence of memory B cells is what makes your body's second encounter with a pathogen so different from the first. The first time you meet a new germ, the immune response is slow. It takes days, sometimes more than a week, for the right B cell to find its match, activate, expand, and produce enough antibodies to control the infection. This is called the primary immune response, and during those early days, you feel it as illness, as fever and fatigue, and the heavy weight of sickness settling into your body. But if that same germ returns, weeks or months or years later, the memory B cells are already there, already primed, already carrying the blueprint for the right antibody. The secondary response is faster by days, stronger by orders of magnitude, and often so swift that the pathogen is eliminated before you ever feel a symptom. You never even know the encounter happened. It is silent protection, invisible defense, the quiet reward of a body that has learned from its own past and carries that learning forward into every future moment. This is the principle behind how vaccines work, and it is one of the most beautiful ideas in all of medicine. A vaccine introduces your immune system to a harmless version of a pathogen, a weakened virus, an inactivated bacterium, a fragment of a protein coat, just enough for your B cells to recognize the antigen, activate, expand, and produce both antibodies and memory cells. Your body goes through the motions of an immune response without the danger of the actual disease. And when it is over, you are left with a gift, a population of memory B cells and long-lived antibodies that will recognize the real pathogen if it ever arrives. The vaccine teaches your body a lesson it never had to learn the hard way. It is a rehearsal, a dress rehearsal for a performance your immune system hopes it will never have to give, but is now perfectly prepared for. And in this gentle rehearsal, in this quiet exchange of information between a needle and a cell, lies the reason that diseases which once devastated entire generations have been reduced to memories. The astonishing diversity of antibodies your body can produce arises from a process that happens deep in the DNA of every developing B cell. The genes that code for antibodies are not single, continuous stretches of DNA like most genes. Instead, they are broken into segments, small pieces labeled V, D, and J. that are scattered across the chromosome like words cut from a sentence and spread across a table. During B cell development, the cell's DNA is physically rearranged. One V segment is chosen at random, one D segment, and one J segment, and they are joined together to form a complete gene. The segments that are not chosen are permanently deleted. This means that each B cell assembles its antibody gene from a unique combination of pieces, a molecular collage that produces a receptor found on no other cell. It is controlled randomness, guided chance, a system that uses the raw material of variation to produce something specific, something personal, something that belongs only to that one cell and the antibodies it will make. This remarkable process of genetic shuffling is called VDJ recombination, and its name, though technical, describes exactly what happens: the joining of variable, diversity, and joining gene segments into a single functional antibody gene. It was first described in detail by Susumu Tonegawa, a Japanese molecular biologist who received the Nobel Prize in Physiology or Medicine in 1987 for this discovery. Before Tonegawa's work, scientists were puzzled by a simple question: how could the body produce millions of different antibodies when the genome contained only a limited number of genes? The answer, Tonegawa showed, was that the body did not need a separate gene for each antibody. It needed only a collection of gene segments and a mechanism for combining them in different ways. It was like discovering that a language does not need a separate symbol for every word. It needs only an alphabet and a set of rules for putting letters together. VDJ recombination is that alphabet, and the antibodies it produces are the words of your immune vocabulary. VDJ recombination is one of the most important reasons your body can produce such an extraordinary range of antibody shapes, but it is not the only one. Additional diversity comes from the imprecise way the gene segments are joined together. Small, random additions or deletions of nucleotides at the junctions between segments, introduced by an enzyme called terminal deoxynucleotidyl transferase. These tiny random changes at the joints between V, D, and J segments are called junctional diversity, and they multiply the number of possible antibody sequences far beyond what the gene segments alone could produce. It is as if, in the process of assembling a sentence from a set of words, you were allowed to add or remove a letter or two at each junction, creating subtle variations in meaning that make each sentence truly unique. This junctional diversity is especially important in the part of the antibody that makes direct contact with the antigen, the region where the fit must be most precise. And so the randomness serves a purpose. It expands the library, deepens the vocabulary, and ensures that somewhere in the trillions of B cells your body produces, there is one whose antibody fits the threat at hand. Even after a B cell has been activated and has begun producing antibodies, the process of refinement does not stop. In a process called somatic hypermutation, the genes encoding the antibody's variable regions undergo rapid, targeted mutations, small changes in the DNA sequence that alter the shape of the antibody's binding site, sometimes for the better and sometimes for the worse. B cells whose mutations produce a tighter fit for the antigen are rewarded. They receive survival signals and continue to multiply. B cells whose mutations produce a worse fit are allowed to die. This is a kind of evolution happening in real time inside your own body, over the course of days rather than millennia. It is natural selection compressed into a single immune response, a process that sculpts and sharpens the antibody's grip on its target with each successive generation. The result is affinity maturation, the gradual improvement in the quality of the antibodies your body produces as the immune response unfolds, a quiet and relentless pursuit of perfection that happens entirely without your awareness. This process of somatic hypermutation and selection takes place in specialized structures called germinal centers, which form inside your lymph nodes during an active immune response. Germinal centers are microscopic arenas of intense activity, places where B cells gather, divide, mutate, and compete for survival in a process that has been compared to a molecular audition. Only the B cells with the best fitting antibodies are selected to survive and become either plasma cells or memory cells. The rest are gently eliminated. Germinal centers were first described in the late 19th century, and their name comes from the German word for seedbed, a place where something new is grown, where potential is cultivated and shaped into form. It is a fitting name because germinal centers are indeed places of growth, of becoming, of transformation. Inside these tiny structures, your immune system is doing something extraordinary. It is learning, in real time, how to build a better antibody, and it is doing so with a patience and precision that would humble any engineer. All of this happens in the warmth of your lymph nodes, in silence while you sleep. One of the simplest and most effective things antibodies can do is clump pathogens together, a process called agglutination. When antibodies bind to antigens on the surface of multiple bacteria or viruses at once, their Y-shaped arms act as bridges, linking one pathogen to another and then another until a cluster forms, a tiny raft of invaders stuck together by the invisible threads of antibody molecules. These clumps are too large to move freely through your tissues, too conspicuous to hide, and far easier for the patrolling cells of your immune system to find and destroy. Agglutination is a strategy of simplicity, of making the invisible visible, of turning scattered enemies into a single, manageable target. It is the molecular equivalent of gathering fallen leaves into a pile, rather than chasing them one by one across a wide and windy field. And it works beautifully, quietly, without any effort from you, happening in the warm currents of your blood and the still spaces of your tissues while you go about your day, or while you rest, as you are resting now. The clumps that antibodies create through agglutination are not just easier to find, they are easier to eat. The cells that consume pathogens in your body, the phagocytes, are drawn to these clusters the way a person might be drawn to a light in a dark room. The clumped pathogens are coated in antibodies, bristling with molecular signals that say, "Here, this is ready, this is prepared for you." And the phagocytes respond with enthusiasm, engulfing the entire clump in a single gulp, wrapping it in a pocket of membrane and flooding it with digestive enzymes that break the pathogens down into harmless fragments. This is the beauty of agglutination. It does not just stop pathogens from spreading, it actively delivers them to the very cells that will destroy them. It is a system of cooperation of one part of the immune system preparing the way for another, a relay of protection that moves through your body with the quiet efficiency of something that has been practiced for hundreds of millions of years. Antibodies can also activate a powerful cascade of proteins called the complement system, and the word complement is well chosen. It means to complete, to make whole, to add what is missing. The complement system is a collection of more than 30 proteins that circulate in your blood in an inactive form, waiting to be triggered. When an antibody binds to an antigen and the stem of the antibody changes shape in just the right way, it sets off a chain reaction among these complement proteins, one activating the next, which activates the next, in a cascade that amplifies the signal with each step. The result is a multi-pronged attack. Some complement proteins punch holes directly in the membranes of bacteria. Others coat the pathogen to make it more appetizing to phagocytes, and still others recruit additional immune cells to the site of infection. It is a system of escalation, of proportional response, triggered by the quiet binding of an antibody to its target, and amplified into something far more powerful than either the antibody or the complement proteins could achieve alone. When the complement cascade reaches its final stage, something remarkable happens. A group of complement proteins assemble together on the surface of a bacterium and form a structure called the membrane attack complex. This complex inserts itself into the bacterium's outer membrane and creates a pore, a hole, a tiny opening that disrupts the careful balance of the cell's interior. Water and ions rush in, the bacterium swells, and eventually it bursts, destroyed not by a single blow but by the slow and steady dismantling of the barrier that kept it alive. It is lysis, the breaking open of a cell, and it is one of the most direct and dramatic consequences of antibody activation. And yet even here there is a kind of gentleness in the mechanism. The membrane attack complex does not tear the bacterium apart. It simply opens a door that was meant to stay closed and lets the natural forces of osmosis and pressure do the rest. It is destruction through exposure, through openness, through the simple and irrevocable act of letting the outside in. When antibodies coat the surface of a pathogen, they transform it into something that the immune system's most voracious cells can recognize instantly. Macrophages and neutrophils, the great eaters of the immune world, carry receptors on their surfaces that are designed to bind to the stems of antibody molecules. When a pathogen is covered in antibodies, these receptors light up and the phagocyte reaches out and engulfs the coated invader with a swift and practiced motion. This is opsonization at work, the process we spoke of earlier, and it is one of the most important functions antibodies perform. Without antibodies, many pathogens are slippery, evasive, difficult for phagocytes to grip. With antibodies, they become conspicuous, adhesive, impossible to ignore. It is as if the antibodies have painted a bright color on something that was trying to hide in shadow, and now every passing immune cell can see it clearly. The partnership between antibodies and phagocytes is one of the oldest and most effective collaborations in immunology, a duet that has been keeping living things safe for longer than there have been creatures with bones. Macrophages are among the most ancient and versatile cells in your immune system, and their name tells you exactly what they do. It comes from the Greek words for large eater. These cells roam through your tissues, patrolling for anything that has been tagged by antibodies or complement proteins, and when they find a coated pathogen, they engulf it completely, drawing it inside themselves in a process called phagocytosis. Once inside the macrophage, the pathogen is enclosed in a small compartment called a phagosome, which then fuses with another compartment filled with powerful digestive enzymes and toxic chemicals. The pathogen is broken down, dismantled, reduced to fragments so small they can be recycled or presented to other immune cells as information. A report on what the invader looked like, a molecular briefing that helps the adaptive immune system refine its response. The macrophage then is not just a destroyer, it is also a teacher, a messenger, a link between the immediate act of consumption and the longer, slower work of learning and remembering. One of the most elegant things antibodies can do is prevent a virus from entering your cells in the first place. Viruses are not truly alive on their own. They are packages of genetic material wrapped in a coat of protein, and they can only reproduce by slipping inside a living cell and hijacking its machinery. To get inside, a virus must first attach to the cell's surface, and it does this using specific proteins on its coat that fit into receptors on the cell like a key fitting into a lock. But if an antibody binds to those viral surface proteins first, it covers the key, blocks the lock, and the virus is left stranded outside the cell, unable to enter, unable to reproduce, unable to do anything at all. This is neutralization, the simplest and perhaps the most beautiful form of immune defense. No inflammation, no destruction, no collateral damage. Just a quiet hand placed over a keyhole, and the virus drifts away harmlessly, like a letter that was never opened, a word that was never spoken. The word neutralization carries within it the idea of balance, of returning something to a state where it can do no harm. When scientists speak of neutralizing antibodies, they mean antibodies that prevent a pathogen from functioning, that strip it of its ability to infect, to spread, to cause disease. Neutralization can happen in several ways. The antibody might block the part of the virus that attaches to cells, or it might change the shape of a toxin so that it can no longer interact with its target, or it might simply coat the pathogen so completely that none of its functional surfaces are exposed. In every case, the principle is the same. The antibody does not destroy the pathogen, but disarms it, renders it inert, returns it to a state of harmlessness. Neutralization is the immune system's most peaceful strategy, its gentlest form of intervention, a defense that works not through violence but through a kind of molecular stillness. The threat is not removed, it is simply made quiet, made small, made unable to be what it once was. When scientists develop a new vaccine and want to know if it is working, one of the most important things they measure is the level of neutralizing antibodies in the blood of vaccinated individuals. This measurement, called a neutralization titer, tells researchers how effectively the antibodies produced by the vaccine can prevent the pathogen from infecting cells in a laboratory setting. A high titer of neutralizing antibodies is one of the strongest indicators that a vaccine is providing protection, and it is used as a benchmark in clinical trials around the world. The search for neutralizing antibodies has driven some of the most important medical research of our time, from the development of vaccines against polio and measles to the rapid creation of COVID-19 vaccines. In every case, the question is the same. Can the antibodies your body makes in response to this vaccine block the pathogen from entering your cells? And when the answer is yes, it means that your body has learned something it will carry forward, a quiet lesson in protection that may last a lifetime. Sometimes an antibody can recognize more than one type of antigen, a phenomenon called cross-reactivity. This happens when two different antigens share a similar shape or a similar molecular feature, similar enough that the antibody's binding site, designed for one, can also grip the other. Cross-reactivity can be a gift or a complication, depending on the circumstances. In some cases, it provides unexpected protection. Antibodies made against one strain of a virus may partially protect against a related strain, offering a kind of borrowed immunity that bridges the gap between two threats. This is part of the reason why exposure to one type of influenza virus can sometimes reduce the severity of infection with a different type. But cross-reactivity can also cause problems. It is one of the mechanisms behind certain autoimmune conditions, where antibodies made against a pathogen happen to recognize a similar structure on the body's own cells. The immune system, in its earnest effort to protect, sometimes mistakes self for other, and the consequences can be as complex as the system itself. There is a particular tenderness in the way antibodies protect the very youngest among us. Newborn babies arrive in the world with immune systems that are still developing, still learning, not yet capable of mounting the full range of responses that an adult's body can produce. But they are not left defenseless. Antibodies in breast milk, particularly IgA, coat the lining of the baby's mouth, throat, and digestive tract with every feeding, creating a protective barrier against the bacteria and viruses the baby encounters in those first vulnerable weeks and months. These antibodies cannot enter the baby's bloodstream through the gut, but they do not need to. They work at the surfaces, at the gates, preventing pathogens from gaining a foothold in the very places where infection is most likely to begin. It is a borrowed immunity, a gift passed from mother to child in the most intimate and ancient of exchanges, and it continues for as long as breastfeeding lasts, adapting and changing as the mother's own immune system encounters new threats in the world they share. The transfer of antibodies from one person to another, whether through the placenta, through breast milk, or through medical treatment, is called passive immunity. The word passive is important here because it distinguishes this kind of protection from the active immunity your body builds through its own encounters with pathogens or vaccines. In passive immunity, the antibodies are not made by the recipient's own immune system. They are received, borrowed, gifted from another source. This means that passive immunity is immediate but temporary. The transferred antibodies will eventually be broken down by the body, and because no memory B cells were created, the protection will fade without a trace. It is protection without learning, defense without memory, a shield that lasts only as long as the borrowed antibodies survive. And yet, for all its temporariness, passive immunity can be life-saving. In newborns who need protection before their own immune systems mature, in patients exposed to deadly toxins, in anyone who needs defense right now today, without the time to wait for the body to learn. One of the most dramatic applications of passive immunity is anti-venom, the treatment used to counteract the effects of venomous bites from snakes, spiders, and other creatures. Anti-venom is made by injecting small, carefully controlled amounts of venom into large animals, usually horses or sheep, and then collecting the antibodies those animals produce in response. These antibodies are purified, concentrated, and prepared for injection into human patients who have been bitten. When administered, the animal-derived antibodies bind to the venom molecules circulating in the patient's blood, neutralizing them before they can cause further damage. It is passive immunity in its most urgent form, borrowed antibodies from another species, crossing the boundary between animal and human to save a life. The history of antivenom stretches back to the late 19th century, when scientists first demonstrated that serum from immunized animals could protect against venom. And even today, more than a century later, antivenom remains one of the most important treatments available for snakebite, a condition that affects millions of people around the world every year. When your body first encounters a new pathogen, the antibody response does not happen instantly. It unfolds over time, like a story finding its rhythm. In the first few days after infection, antibody levels in the blood remain low as the immune system identifies the threat, activates the right B cells, and begins the process of clonal expansion. IgM is the first to appear, rising in the blood within the first week, a broad and urgent response that buys the body time. Then, gradually, IgG begins to rise, increasing steadily until it reaches its peak roughly 1 to 2 weeks after the infection began. This peak represents the full force of the adaptive immune response, a flood of specific, high-quality antibodies pouring into the blood from the plasma cells that have been working day and night in the lymph nodes and bone marrow. After the infection is cleared, antibody levels decline, but they do not return to zero. A baseline of protective antibodies remains, a quiet residue of the battle, a molecular scar that marks where the body fought and won. And beneath that scar, the memory cells wait, ready to respond again if the need ever arises. Right now, as you lie here and listen, as your muscles soften and your thoughts begin to drift, millions of antibodies are circulating through your body, moving through your bloodstream with the quiet persistence of a river that never stops flowing. They are in the blood that runs through the veins of your arms and legs, in the capillaries that thread through your lungs, in the small vessels that feed the tissue behind your eyes and the skin of your fingertips. They are in the clear fluid of your lymph, moving slowly through a parallel system of channels and nodes that drains and filters the spaces between your cells. They are everywhere, and they are always working, always scanning, always comparing the shapes they encounter against the memory of shapes they were built to recognize. You do not have to direct them. You do not have to think about them. They are the most faithful workers in the most silent factory you will ever own, and they are doing their job right now, right here, in this very moment, as you begin to let go of the day and sink deeper into rest. Your tears are more than just salt water and emotion. They are a carefully composed fluid that includes, among its many components, IgA antibodies that help protect the delicate surface of your eyes. Every time you blink, a thin film of tear fluid is spread across the cornea, and within that film are antibodies that bind to bacteria and viruses that might otherwise settle on the surface and cause infection. Your eyes are one of the most exposed and vulnerable parts of your body. They are open windows to the world, portals through which light enters and images are formed, and they are constantly in contact with the air and everything the air carries. The antibodies in your tears are part of the quiet army that guards these windows, keeping them clear, keeping them clean, working in the thin layer of moisture that keeps your vision sharp and your eyes comfortable. You blink, and in that blink, antibodies are spread across the surface of your sight. You blink again, and with each blink, the protection is renewed, as faithfully as the turning of the tide. Your saliva, too, is a carrier of protection. Every time you breathe through your mouth, every time you swallow, every time you speak or yawn or simply rest with your lips parted, the IgA antibodies in your saliva are there, coating the inside of your mouth and the back of your throat with a thin and invisible shield. The mouth is one of the body's busiest borders, a place where food enters, where air passes, where billions of bacteria live in communities on your tongue and gums and teeth. Most of those bacteria are harmless, even helpful, but among them there are always a few that could cause problems if they were allowed to multiply unchecked. The IgA in your saliva keeps them in balance, binding to potential pathogens and preventing them from attaching to the cells that line your oral cavity. It is a quiet negotiation, a gentle maintenance of order at one of the most active crossroads in your body, and it happens continuously, hour after hour, without a single conscious thought from you. The mucous membranes of your nose are lined with a rich layer of antibodies, part of the body's first line of defense against the countless particles and pathogens you inhale with every breath. Each time air enters your nostrils, it passes over a warm, moist surface coated with mucus, a thick, sticky fluid that traps dust, pollen, bacteria, and viruses before they can travel deeper into your lungs. Within that mucus, IgA antibodies are waiting, ready to bind to any pathogen that becomes trapped. The antibodies do not kill the pathogens themselves. They simply hold them, coat them, prevent them from attaching to the cells of your nasal lining, and the mucus itself carries them away, swept toward the back of your throat by the rhythmic beating of tiny hair-like structures called cilia. It is an extraordinary system of filtration and defense happening with every breath you take, cleaning the air before it reaches the delicate tissues of your lungs, and you have never once had to think about it. Your nose is not just a passage for air, it is a guardhouse staffed with molecular sentinels, and it has been protecting you since your very first breath. Colostrum, the thick golden fluid that a mother's breasts produce in the first few days after giving birth before the mature milk comes in, is one of the most concentrated sources of antibodies found anywhere in nature. It is rich in IgA, and it contains higher concentrations of immune proteins than any breast milk that will follow. Colostrum is sometimes called liquid gold, not just for its color but for its extraordinary value to a newborn whose immune system is still fragile, still learning, still months away from being able to mount its own robust defense. When a baby swallows colostrum, the antibodies it contains coat the lining of the baby's gut and respiratory tract, providing immediate protection against the bacteria and viruses the baby encounters in its first hours and days of life. It is the mother's immune system reaching across the boundary of two bodies to shield the one that is most vulnerable, and it is one of the most beautiful examples of biological generosity in the natural world, a gift of protection offered in the very first feeding at the very beginning of a new life. Your blood type, the classification that determines whether you are type A, type B, type AB, or type O, is determined in part by which antibodies are present in your plasma. If you have type A blood, your red blood cells carry A antigens on their surface and your plasma contains anti-B antibodies, antibodies that would attack type B red blood cells if they were ever introduced into your bloodstream. If you have type B blood, the reverse is true, B antigens on your cells, anti-A antibodies in your plasma. If you are type AB, you carry both antigens and neither antibody. And if you are type O, you carry neither antigen but both antibodies. This system of natural antibodies is one of the reasons blood transfusions must be carefully matched. If the wrong type of blood is given, the recipient's antibodies will bind to the foreign red blood cells, clumping them together and triggering a potentially life-threatening reaction. Your blood type antibodies are always there, always circulating, a permanent part of your blood's identity, and they have been with you since the first months of your life. People with type O blood occupy a special place in the story of antibodies and blood types. Their red blood cells carry neither A nor B antigens, which means their blood can be given to people of any blood type without triggering an antibody-mediated rejection. This is why type O negative blood is called the universal donor type, the blood that emergency rooms reach for when there is no time to test. But in their own plasma, people with type O blood carry both anti-A and anti-B antibodies, a double guard that would react against any red blood cells carrying either antigen. This means that while they can give blood to almost anyone, they can only safely receive blood from other type O donors. It is a curious asymmetry, a generosity built into the blood itself, the ability to give widely, paired with the need to receive narrowly. And it is all governed by antibodies, those small and specific molecules that float in the plasma, quietly enforcing the rules of compatibility that make safe transfusion possible, rules that have saved countless lives since the discovery of blood types in the early 20th century. If you have ever sneezed in the presence of pollen, or felt your eyes water near a cat, or watched your skin redden after touching something that disagreed with you, then you have felt the work of IgE antibodies in their most overzealous form. Allergic reactions are, at their core, cases of mistaken identity, your immune system encountering a harmless substance and treating it as though it were a dangerous invader. The process begins when IgE antibodies, sensitized by a previous exposure to the allergen, sit on the surface of mast cells in your tissues, waiting. When the allergen appears again, a grain of pollen landing on the lining of your nose, a molecule of peanut protein touching the wall of your gut. It binds to the IgE, and the mast cell is triggered to release a flood of inflammatory chemicals, including the histamine that causes swelling, itching, sneezing, and all the familiar discomforts of an allergic response. It is protection without a threat, defense without a danger, and it is one of the most common ways that the immune system's extraordinary sensitivity can become, for a moment, a burden rather than a gift. Histamine is the chemical most directly responsible for the symptoms you feel during an allergic reaction, and its release is triggered by the binding of an allergen to IgE antibodies on the surface of mast cells. Mast cells are found throughout your body's tissues, particularly in the places where you most frequently encounter the outside world: your skin, your lungs, your nose, your digestive tract. They are packed with granules, tiny containers filled with histamine and other inflammatory molecules, ready to release their contents at a moment's notice. When an allergen cross-links the IgE molecules on a mast cell's surface, bridging two or more IgE antibodies with a single allergen molecule, The mast cell erupts, releasing its granules in a rapid burst called degranulation. The histamine that floods out causes blood vessels to dilate, tissues to swell, mucus to be produced, and nerves to fire, creating the itching, sneezing, watery eyes and congestion that are so familiar to anyone who lives with allergies. It is a response built for emergencies that is sometimes triggered by ordinary things, and this is why antihistamines, drugs that block the effects of histamine, are among the most widely used medications in the world. Sometimes the immune system makes a mistake that is both profound and deeply personal. It produces antibodies that recognize not a foreign invader, but the body's own tissues. These are called autoantibodies, and their presence is the hallmark of autoimmune disease. The word auto means self, and an autoantibody is, in the simplest sense, an antibody that has turned inward, that has lost the ability to distinguish between what belongs and what does not. How this happens is not entirely understood, but it likely involves a combination of genetic predisposition, environmental triggers, and the occasional failure of the checkpoints that normally prevent self-reactive B cells from maturing and entering the bloodstream. The result is a body at odds with itself, an immune system that attacks the very tissues it was designed to protect. It is one of the most poignant failures in all of biology, a system of extraordinary precision undone by a single error of recognition. And yet, even in autoimmune disease, the underlying machinery is the same. The same B cells, the same antibodies, the same elegant specificity, directed not at an enemy, but at a friend. Autoantibodies play a central role in many of the autoimmune conditions that affect millions of people around the world. In lupus, systemic lupus erythematosus, autoantibodies target components of the cell nucleus, including DNA itself, leading to widespread inflammation that can affect the skin, joints, kidneys, heart, and brain. In rheumatoid arthritis, autoantibodies called rheumatoid factor and anti-CCP antibodies target the lining of the joints, causing the chronic pain, swelling, and stiffness that characterize the disease. In type 1 diabetes, autoantibodies attack the insulin-producing cells of the pancreas, destroying the body's ability to regulate blood sugar. In each case, the mechanism is the same. Antibodies that have lost their way, that have turned their extraordinary specificity against the self. These conditions are reminders that the immune system, for all its sophistication, is not infallible, and that the line between protection and harm can sometimes be as thin and delicate as the fold of a protein. Understanding autoantibodies has become one of the great challenges and promises of modern medicine, and the search for ways to redirect them continues in laboratories and clinics around the world. Here is something worth knowing on a night like this. Stress and sleep deprivation can reduce your body's ability to produce antibodies. When you are under chronic stress, your body produces elevated levels of cortisol, a hormone that, among its many effects, suppresses the activity of the immune system. Prolonged exposure to cortisol can reduce the number and activity of B cells, impair the function of helper T cells that B cells depend on for activation, and diminish the overall antibody response to infections and vaccines. Sleep deprivation has similar effects. Studies have shown that people who sleep fewer than 6 hours a night produce significantly fewer antibodies in response to vaccination than people who get a full night's rest. Your immune system, it turns out, needs rest just as much as your muscles and your mind do. It needs the quiet hours of sleep to consolidate its responses, to refine its antibodies, to do the slow and careful work of memory and repair. And so, in a very real and biological sense, the act of resting right now, of letting your body settle into stillness, is one of the best things you can do for the tiny guardians that live inside you. Research has shown, with a clarity that is both reassuring and beautiful, that restful sleep improves the antibody response to vaccines. In one well-known study, participants who slept a full night after receiving a hepatitis A vaccine produced more than twice as many antibodies as those who stayed awake. Other studies have found similar results with influenza vaccines. Good sleep in the days surrounding vaccination is associated with higher antibody titers and longer-lasting protection. The reasons are not fully understood, but scientists believe that sleep provides a unique environment for the immune system to do its most important work. During deep sleep, the body releases growth hormone, reduces cortisol, and shifts the balance of immune signaling in ways that favor antibody production and memory cell formation. It is as if sleep is the quiet workshop where the immune system does its finest crafting, shaping the antibodies and memory cells that will protect you in the weeks and months ahead. So tonight, as you drift towards sleep, know that you are not just resting your body and your mind, you are giving your immune system the time and space it needs to build something extraordinary. In the middle of the 20th century, scientists learned to do something extraordinary. They learned to make copies of a single antibody, identical in every way, over and over again, in limitless supply. These are called monoclonal antibodies, and the word monoclonal means exactly what it sounds like. From a single clone, from one line of cells, all producing the same antibody with the same specificity, the same shape, the same purpose. Before this technology existed, scientists could only harvest antibodies from the blood of immunized animals, a messy, variable process that yielded a mixture of many different antibodies, each one recognizing a slightly different part of the antigen. Monoclonal antibodies changed everything. They gave researchers a tool of unprecedented precision, a way to target a single molecule with perfect specificity, and they opened the door to an entirely new era of medicine. Today, monoclonal antibodies are used to treat cancers, autoimmune diseases, infections, and more, and they are among the most important therapeutic tools ever developed. Their story is a story of human ingenuity in conversation with the immune system's ancient wisdom. The medical applications of monoclonal antibodies are vast and still expanding, a testament to the extraordinary versatility of the antibody molecule itself. In cancer treatment, monoclonal antibodies are designed to recognize specific proteins on the surface of tumor cells, flagging them for destruction by the patient's own immune system or delivering toxic drugs directly to the cancer while sparing healthy tissue. In autoimmune diseases, monoclonal antibodies can block the inflammatory molecules that drive the condition, calming the overactive immune response and reducing symptoms. In infectious disease, monoclonal antibodies can neutralize viruses and toxins with a precision that few other treatments can match. There are now more than 100 monoclonal antibody therapies approved for use around the world, and hundreds more are in development. Each one is a product of the same basic insight, that the antibody's natural ability to recognize and bind to specific targets can be harnessed, refined, and directed by human hands to treat diseases that were once considered untreatable. It is medicine guided by the quiet intelligence of the immune system itself. The first monoclonal antibody therapy approved for clinical use was murimonerb, known as OKT3, and it was approved in 1986 for the prevention of organ transplant rejection. It worked by targeting a protein on the surface of T cells, the immune cells responsible for attacking the transplanted organ, and suppressing their activity long enough for the body to accept the new organ. It was a groundbreaking moment, the first time a laboratory-made antibody was used as a medicine in a human patient, and it opened a door that has never closed. In the years since, the technology has advanced enormously. From mouse-derived antibodies that sometimes triggered immune reactions in human patients, to humanized and fully human monoclonal antibodies that the body accepts with far fewer side effects. Each generation of monoclonal antibody therapy has been more refined, more specific, more gentle than the last, and the field continues to evolve with a speed and creativity that mirrors the immune system it draws inspiration from. What began with a single approval in 1986 has become one of the most dynamic and promising areas of modern medicine. The technique that made monoclonal antibodies possible was developed in 1975 by two scientists working at the Medical Research Council Laboratory of Molecular Biology in Cambridge, England. Their names were César Milstein, an Argentine-British biochemist, and Georges Köhler, a German biologist, and together they invented a method for producing a single type of antibody in unlimited quantities. The technique involved fusing an antibody-producing B cell with a cancer cell called a myeloma, creating a hybrid cell, a hybridoma, that had the B cell's ability to make a specific antibody and the cancer cell's ability to divide indefinitely. The hybridoma could be grown in culture, maintained in the laboratory, and used to produce a never-ending supply of identical antibodies. It was an elegant solution to a problem that had frustrated immunologists for decades, and it transformed not only medicine but also research, diagnostics, and biotechnology. Milstein and Köhler published their method in the journal Nature, and the technique they described remains in its essential principles the foundation of monoclonal antibody production to this day. In 1984, César Milstein and Georges Köhler were awarded the Nobel Prize in Physiology or Medicine for their development of the hybridoma technique and its contribution to the field of monoclonal antibodies. They shared the prize with Niels Kai Jerne, a Danish immunologist whose theoretical work on the immune system had laid much of the intellectual groundwork for understanding how antibodies are produced and regulated. The Nobel Committee recognized that the ability to produce monoclonal antibodies had opened an entirely new chapter in biology and medicine, a chapter that is still being written today. Milstein, who had fled the political turmoil of Argentina to build his career in England, was known for his generosity and his insistence that scientific knowledge should be shared freely. He and his colleagues did not patent the hybridoma technique, a decision that allowed it to spread rapidly through laboratories around the world and accelerate the development of monoclonal antibody therapies. It was a gift in its own way, not unlike the antibodies themselves, something precise, something powerful, offered freely for the benefit of all. One of the most innovative applications of monoclonal antibodies in medicine is the antibody-drug conjugate, a hybrid molecule that combines the targeting precision of an antibody with the cell-killing power of a chemotherapy drug. The idea is elegant in its simplicity. Attach a toxic drug to an antibody that recognizes a specific protein on the surface of cancer cells, and the antibody will carry the drug directly to the tumor, delivering it precisely where it is needed while sparing the rest of the body. Once the antibody binds to the cancer cell, the entire conjugate is drawn inside the cell and the drug is released, killing the cancer from within. It is targeted therapy in its most literal sense, a missile guided not by technology but by the same ancient mechanism of molecular recognition that your immune system has been using for hundreds of millions of years. Antibody-drug conjugates are now approved for the treatment of several types of cancer, and dozens more are in clinical trials, each one a testament to the idea that the antibody's natural wisdom can be amplified and directed by human creativity. If you have ever taken a home pregnancy test, you have held in your hands a device built on the remarkable specificity of antibodies. Pregnancy tests work by detecting the presence of a hormone called human chorionic gonadotropin, or HCG, which is produced by the placenta shortly after a fertilized egg implants in the wall of the uterus. The test strip contains antibodies that are designed to bind specifically to HCG molecules. When a sample of urine is applied to the strip, any HCG present binds to these antibodies, triggering a visible signal, usually a colored line, that indicates pregnancy. The entire process takes only minutes, and it is possible because of the extraordinary precision of antibody-antigen recognition, the ability of an antibody to detect a single specific molecule among the thousands of other proteins and chemicals present in a drop of urine. It is one of the most widely used medical tests in the world, and at its heart, it is a conversation between an antibody and a hormone, a molecular handshake that carries with it the weight of life-changing news. Many of the rapid diagnostic tests that became familiar to millions of people during the COVID-19 pandemic rely on the same basic principle, the ability of antibodies to detect specific molecules with extraordinary precision. Rapid antigen tests, which detect viral proteins in a nasal swab, use antibodies fixed to a test strip that bind to proteins from the surface of the SARS-CoV-2 virus. If the viral proteins are present in the sample, they are captured by the antibodies, and a visible signal appears. Antibody tests, on the other hand, detect whether a person's own immune system has produced antibodies against the virus, a sign of past infection or vaccination. Both types of tests owe their speed and simplicity to the fundamental properties of antibodies, their specificity, their ability to bind tightly to their targets, and their capacity to be manufactured and deployed in forms that are stable, portable, and accessible. In a time of global uncertainty, it was the ancient molecule of the antibody, repurposed, mass-produced, and placed in the hands of ordinary people, that provided one of the most important tools for understanding and managing the spread of disease. Serology is the branch of science devoted to the study of blood serum, the clear yellowish fluid that remains after blood has clotted and the cells have been removed. It is in this serum that antibodies are found in their highest concentrations, and serological tests have been a cornerstone of medicine and public health for more than a century. By measuring the types and amounts of antibodies in a person's serum, doctors can determine whether a person has been exposed to a particular infection, whether a vaccine has produced an adequate immune response, and whether the immune system is functioning properly. Serology can also help diagnose autoimmune diseases, identify blood types for transfusion, and screen donated blood for dangerous pathogens. The field was born in the late 19th century when scientists first discovered that the serum of immunized animals contained protective factors that could be transferred to others, and it has grown into a vast and essential discipline that touches nearly every corner of modern medicine. Every time blood is drawn and tested for antibodies, it is serology at work. The quiet science of reading the immune history written in a drop of your blood. One of the most widely used methods for measuring antibodies in the laboratory is a technique called ELISA, enzyme-linked immunosorbent assay. Despite its technical name, the principle behind ELISA is wonderfully simple. A surface, usually a small plastic well, is coated with the antigen of interest. A sample of the patient's blood serum is added, and if antibodies specific to that antigen are present, they will bind to the surface. After washing away everything that did not stick, a second antibody, one that recognizes human antibodies and is linked to an enzyme, is added. This second antibody binds to any patient antibodies that are present, and when a chemical substrate is added, the enzyme produces a color change that can be measured. The darker the color, the more antibodies were present in the sample. ELISA is used in hospitals and research laboratories around the world, millions of times every day, to detect everything from HIV to food allergens to hormones. It is a testament to the principle that the best tools are often built on the simplest ideas, and that the antibody's natural talent for recognition can be harnessed in ways that are both elegant and profoundly useful. Therapeutic antibodies, antibodies designed and manufactured as medicines, are now one of the fastest growing categories in the pharmaceutical industry. The global market for antibody-based therapies is worth hundreds of billions of dollars and continues to expand as new targets are identified and new technologies make it possible to design antibodies with greater precision than ever before. This growth reflects a fundamental shift in the way medicine is practiced, a move away from small molecule drugs that affect the entire body toward targeted therapies that interact with specific molecules in specific places, minimizing side effects and maximizing efficacy. Antibodies are ideally suited to this approach because of their natural specificity, their ability to be engineered and modified in the laboratory, and their compatibility with the human body. They can be designed to last longer in the blood, to trigger or suppress specific immune responses, to carry drugs or radioactive isotopes to precise targets, and to be produced at scale in bioreactors. The antibody, once a molecule known only to immunologists, has become one of the most important and versatile platforms in all of modern pharmacology. Among the many monoclonal antibody therapies that have transformed medicine, rituximab holds a special place. Approved in 1997, rituximab was one of the first monoclonal antibodies developed for the treatment of cancer, and it changed the prognosis for patients with certain types of non-Hodgkin lymphoma, a group of cancers that arise from lymphocytes, the very immune cells that produce antibodies. Rituximab targets a protein called CD20, which is found on the surface of B cells, and when it binds, it triggers the destruction of those cells by the patient's own immune system. Because the cancerous B cells in non-Hodgkin lymphoma express CD20 on their surface, rituximab can selectively target them while leaving other types of immune cells relatively unharmed. The impact of rituximab has been profound. It has improved survival rates, reduced the need for aggressive chemotherapy, and become a standard part of treatment for several types of lymphoma. It has also been approved for use in rheumatoid arthritis and other autoimmune conditions, extending its reach far beyond cancer and demonstrating the remarkable versatility of monoclonal antibody therapy. In recent years, antibody therapies have been explored as a potential treatment for Alzheimer's disease, one of the most devastating and poorly understood conditions of aging. Alzheimer's is characterized by the accumulation of abnormal protein deposits in the brain, particularly amyloid beta plaques and tau tangles, that are associated with the progressive loss of memory, cognition, and identity. Researchers have developed monoclonal antibodies designed to bind to amyloid beta and help clear these plaques from the brain, and several of these antibodies have entered clinical trials with results that have sparked both hope and careful debate within the scientific community. The challenge is immense. The brain is protected by the blood-brain barrier, which limits the entry of large molecules like antibodies, and the relationship between amyloid plaques and cognitive decline is more complex than once thought. But the effort continues, driven by the possibility that the antibody's ancient talent for recognition and clearance might be directed against one of the greatest medical challenges of our time. It is a frontier where immunology meets neuroscience, where the molecule of protection reaches toward the mystery of the mind. Antibodies are not unique to humans. They are found in all jawed vertebrates, a vast group of animals that includes every fish with a jaw, every amphibian, every reptile, every bird, and every mammal on Earth. From the great white shark gliding through dark ocean currents to the sparrow perched outside your window. From the frog resting on a damp stone to the elephant standing in golden light. Every one of these creatures carries antibodies in its blood, uses them to fight infection, and relies on the same basic principles of antigen recognition and immune memory that your own body uses. The antibody-based immune system is one of the great shared inheritances of vertebrate life, a system so successful, so adaptable, that it has been maintained and refined across hundreds of millions of years of evolution, surviving mass extinctions, ice ages, and the relentless pressure of an ever-changing microbial world. When you think of your antibodies, you are thinking of something that connects you to every creature with a backbone, a molecular thread that runs through the entire tapestry of vertebrate life. Sharks are among the most ancient creatures on Earth to possess an antibody-based immune system, and their lineage stretches back more than 400 million years, long before the first dinosaur walked, long before the first flower bloomed. The immune system of sharks contains antibodies that are recognizably similar to our own, with the same basic Y-shaped structure, the same principles of antigen recognition, and the same mechanisms of genetic recombination that generate diversity. One type of shark antibody, called IgNAR, is of particular interest to scientists because it is unusually small and stable, composed of only heavy chains without the light chains found in human antibodies. This makes IgNAR molecules compact and robust, able to withstand conditions that would denature larger, more fragile antibodies. Researchers are studying shark antibodies for potential medical applications, drawn by their stability and their ability to bind to targets that are difficult for human antibodies to reach. The shark, that silent traveler of the deep, carries within its blood a form of molecular wisdom that science is only now beginning to understand and appreciate. Camels, llamas, and their relatives, the members of the camelid family, produce a type of antibody that is found nowhere else in the mammalian world. Alongside their conventional antibodies, camelids make a special class of antibodies that are composed entirely of heavy chains, without the light chains that are normally part of every antibody molecule. The antigen binding region of these heavy chain-only antibodies is remarkably small, just a single domain, about 1/10 the size of a conventional antibody, and scientists have named these tiny binding fragments nanobodies. Nanobodies are extraordinarily stable, resistant to heat and chemical degradation, and small enough to reach molecular targets that larger antibodies cannot access, such as the deep grooves and hidden pockets on the surface of viruses and enzymes. Since their discovery in 1993, nanobodies have become one of the most exciting tools in biotechnology, with applications in diagnostics, imaging, and therapy. It is one of the most charming surprises in immunology that some of the smallest and most powerful antibody fragments in nature come from some of the largest and most gentle animals on Earth. The medical and research applications of nanobodies are growing rapidly, driven by their unique combination of small size, high stability, and ease of production. Because they are composed of just a single protein domain, nanobodies can be produced in bacteria and yeast, organisms that are far simpler and cheaper to grow than the mammalian cell lines required for conventional antibody production. This makes them potentially more accessible and affordable, particularly for use in low-resource settings. Nanobodies are being developed as treatments for a wide range of conditions, including inflammatory diseases, cancers, and viral infections. One nanobody-based therapy, caplacizumab, has already been approved for the treatment of a rare blood clotting disorder called acquired thrombotic thrombocytopenic purpura. Others are in clinical trials for diseases ranging from respiratory syncytial virus to certain solid tumors. In research, nanobodies are used as tools for imaging individual proteins inside living cells, for studying the structure of molecules, and for creating new types of biosensors. The story of nanobodies is a reminder that some of the most powerful innovations in science come not from human invention alone, but from paying close attention to the solutions that nature has already found. Chickens have their own extraordinary way of passing immunity to their young. They concentrate antibodies in the yolks of their eggs. The primary antibody class found in egg yolk is called IgY, which is the avian equivalent of mammalian IgG. As the egg develops inside the hen, IgY antibodies from the mother's bloodstream are actively transported into the growing yolk, accumulating there in high concentrations. When the chick develops inside the egg, it absorbs these antibodies from the yolk, gaining passive immunity against the infections the mother has encountered during her life. It is a system analogous to the transfer of IgG across the placenta in mammals, but adapted to the unique biology of egg-laying creatures. Scientists have taken advantage of this natural process by immunizing hens against specific pathogens and then harvesting the IgY antibodies from their eggs, a method that is less invasive than drawing blood from mammals and that can produce large quantities of antibodies at relatively low cost. Egg-derived antibodies are being explored for use in diagnostics, research, and even as oral therapies for certain gastrointestinal infections. The humble egg, it turns out, is a vessel of immune inheritance, as rich in protection as it is in nourishment. The concept of immunity, the idea that surviving a disease can protect you from getting it again, was observed and written about for centuries before anyone knew that antibodies existed. The ancient Greek historian Thucydides, writing about the plague of Athens in 430 BCE, noted that those who had recovered from the disease were able to care for the sick without falling ill a second time. In medieval China and the Ottoman Empire, practitioners developed a technique called variolation, in which material from a mild smallpox lesion was deliberately introduced into a healthy person's skin, causing a mild infection that conferred lasting protection against the more dangerous form of the disease. And in 1796, the English physician Edward Jenner demonstrated that inoculation with cowpox, a related but milder disease, could protect against smallpox, a discovery that laid the foundation for the science of vaccination. All of these observations and practices were, in essence, attempts to harness the power of immune memory, the ability of the body to learn from experience and carry that learning forward. They were reaching for the antibody without knowing its name, guided by the intuition that the body had its own quiet form of wisdom. The modern scientific story of antibodies begins in 1890, with a discovery that would change the course of medicine forever. In that year, a German physiologist named Emil von Behring, working alongside the Japanese bacteriologist Kitasato Shibasaburo, demonstrated that the blood serum of animals immunized against diphtheria contained a protective factor, something that could neutralize the diphtheria toxin and, when transferred to another animal, protect it from the disease. They called this protective factor antitoxin, and its discovery was a revelation. It showed for the first time that the blood itself could carry specific, transferable protection against disease. Von Behring quickly applied this discovery to the treatment of human patients, developing diphtheria antitoxin therapy that saved the lives of countless children during a time when diphtheria was one of the leading causes of childhood death. The antitoxin was not yet understood to be an antibody, but that is exactly what it was. A molecule of exquisite specificity, born from the immune response circulating in the serum, ready to neutralize a threat it had been trained to recognize. For his discovery of diphtheria antitoxin and its application to the treatment of disease, Emil von Behring was awarded the very first Nobel Prize in Physiology or Medicine in 1901. The Nobel Committee recognized that his work had placed in the hands of physicians a victorious weapon against illness and death, a high praise for a discovery that was, at its heart, about the protective power of the blood itself. Von Behring's work opened an entirely new field of medicine, one based on the principle that the immune system's own products could be harnessed and used as treatments. In the years that followed, antitoxins and antisera were developed for a range of diseases, from tetanus to botulism, and the search for the specific molecules responsible for this protection, the antibodies, became one of the great quests of 20th century science. Von Behring did not live to see the antibody molecule fully characterized, but the path he set in motion led directly to the discoveries that followed. His Nobel Prize was not just a recognition of one man's work. It was the opening note of a symphony that is still being played, more than a century later. The word antibody was first used in the scientific literature in the late 1800s, and it was derived from the German word Antikörper, a compound of anti meaning against, and Körper meaning body or substance. The term was coined to describe the mysterious protective factors found in the blood serum of immunized animals, the substances that could bind to toxins and pathogens and neutralize their effects. In those early years, the nature of these antibodies was entirely unknown. They were defined only by what they did, not by what they were. Scientists debated whether they were chemical substances, cellular products, or something else entirely. It would take decades of research, decades of careful biochemistry, protein purification, and structural analysis before the antibody was revealed to be a protein, a Y-shaped immunoglobulin produced by B cells with a structure that could be described, a function that could be explained, and a diversity that could take the breath away. The name antibody has survived all these discoveries intact, a simple and enduring label for one of the most complex and beautiful molecules in all of biology. The structure of the antibody molecule, the actual physical arrangement of its atoms, its chains, its domains, its Y-shaped form, was first described in detail by two scientists working independently on different sides of the Atlantic. Gerald Edelman, an American biochemist at Rockefeller University, and Rodney Porter, a British biochemist at the University of Oxford, each approached the problem from a different angle. Porter used enzymes to cleave the antibody molecule into fragments, revealing that it could be divided into two functional parts: the arms of the Y, which bind antigens, and the stem, which communicates with the rest of the immune system. Edelman took a more complete approach, breaking the antibody into its individual protein chains and determining their full amino acid sequences, ultimately revealing that the molecule is composed of 4 chains, 2 heavy and 2 light, held together by disulfide bonds. Together, their work painted the first complete portrait of the antibody molecule, revealing its elegant architecture and explaining how its structure gives rise to its function. It was a triumph of molecular biology, a moment when the invisible became visible and the mysterious became understood. In 1972, Gerald Edelman and Rodney Porter were jointly awarded the Nobel Prize in Physiology or Medicine for their discoveries concerning the chemical structure of antibodies. The award recognized that their work had solved one of the most fundamental mysteries in immunology, the question of what an antibody actually is at the molecular level. By revealing the 4-chain structure of the antibody, the distinction between heavy and light chains, and the relationship between the molecule's shape and its function, Edelman and Porter provided the foundation on which all subsequent antibody research has been built. Their discoveries made it possible to understand how antibodies bind to antigens, how the immune system generates diversity, and how antibodies can be engineered and modified for medical use. The Nobel Prize was a recognition not just of their individual achievements, but of the profound importance of the antibody molecule itself. A molecule that, once its structure was known, revealed itself to be one of the most versatile, elegant, and powerful tools in all of biology. Everything that followed, monoclonal antibodies, antibody engineering, immunotherapy, was made possible by the structural understanding that Edelman and Porter provided. Not all vertebrates use antibodies. The most ancient lineages of vertebrates, the jawless fish, including lampreys and hagfish, lack the conventional antibody-based immune system entirely. Instead, they have evolved a completely independent system of adaptive immunity based on different molecules called variable lymphocyte receptors, or VLRs. These receptors perform a function similar to antibodies. They recognize and bind to specific foreign molecules, but they are built from an entirely different set of molecular building blocks, using a different type of structural diversity to achieve the same end. It is a stunning example of convergent evolution, of nature arriving at the same solution through 2 completely different paths. The discovery of VLRs in the early 2000s revealed that the need for adaptive immunity, the ability to learn, to remember, to respond specifically to individual threats, is so fundamental that evolution invented it not once but twice in two separate lineages, hundreds of millions of years apart. Lampreys and humans, separated by nearly 500 million years of evolution, each found their own way to build a system of molecular memory. It is a quiet testament to the depth and urgency of the need to remember, to recognize, to protect. Your body began producing its own antibodies before you were born, while you were still floating in the warm darkness of the womb. By the second trimester of pregnancy, a developing fetus has already begun to produce IgM, the first and most ancient class of antibody, the one that does not cross the placenta and therefore cannot be borrowed from the mother. This means that some of the earliest acts of your immune system happened in a world you have no memory of, in a body so small it could have rested in the palm of a hand. The fetus does not produce antibodies in large quantities, and its immune system is still profoundly immature. But the machinery is there, the B cells are present, and the first tentative steps of immune development are already underway. It is one of the most astonishing facts of human biology that even before you took your first breath, even before your eyes opened to the light, your body was already beginning the lifelong work of learning to protect itself. You arrived in the world not as a blank page but as a story already begun, with the first sentences already written in the language of antibodies. By the time you are a few years old, you carry within you a library of memory, B cells shaped by every germ you have ever encountered, every cold virus that gave you a runny nose, every stomach bug that kept you home from school, every bacterium that slipped past the barriers of your skin and was quietly defeated before you ever knew it was there. Each of these encounters left behind a trace, a molecular memory, a population of B cells that will remember the shape of that particular invader for years, perhaps decades, perhaps a lifetime. This library grows with every passing year, expanding with each new infection and each new vaccination, building a catalogue of immune memory that is uniquely your own. No two people carry exactly the same library because no two people have lived exactly the same immunological life. Your library reflects your personal history, where you have lived, what you have eaten, who you have been close to, which parts of the microbial world you have encountered. It is a biography written not in words but in the shapes of molecules, and it is one of the most intimate and personal things about you. Antibodies can evolve within your own lifetime, becoming more precise with each encounter with the same pathogen. This is what somatic hypermutation and affinity maturation achieve, a process of Darwinian selection happening not across generations of organisms, but within the span of a single immune response, inside the germinal centers of your lymph nodes. The first antibodies your body produces against a new threat are imperfect, approximate, good enough to begin the fight but far from their best. With each subsequent exposure, the antibodies improve, their binding sites refined by mutation and selection until they grip their target with an almost perfect fit. This means that the antibody you produce the second time you encounter a virus is better than the one you made the first time, and the one you make the third time is better still. It is evolution in miniature, compressed into days and weeks instead of millennia, a process that makes your immune system not just reactive but truly adaptive, capable of learning, of improving, of growing wiser with time. Your antibodies are not static, they are living works in progress, always becoming more than they were. The bond between an antibody and its antigen is one of the strongest non-covalent interactions found anywhere in nature. Non-covalent means that the bond does not involve the sharing of electrons between atoms. It is not the kind of bond that holds the atoms of a water molecule together. Instead, the antibody-antigen bond is formed by a constellation of weaker forces, hydrogen bonds, electrostatic attractions, van der Waals forces, and hydrophobic interactions, all working together across the broad surface where the antibody and antigen meet. Individually, each of these forces is modest, but collectively, when dozens or hundreds of them align across a perfectly complementary interface, they create a bond of remarkable strength and specificity. The antibody grips its antigen with a tenacity that belies the gentleness of the individual forces involved, holding on through the turbulence of the bloodstream, through changes in temperature and acidity, through the jostling of countless other molecules. It is a bond built not from a single mighty connection, but from the cooperation of many small ones. A lesson, perhaps, that the greatest strength is sometimes found not in a single great force, but in the quiet accumulation of many gentle ones. some antibodies remain in your body for a lifetime after a single vaccination. This extraordinary longevity is one of the great achievements of the immune system, and it is maintained by long-lived plasma cells that take up residence in the bone marrow, that same warm, hidden tissue where B cells are born, and continue to produce antibodies at a low, steady rate for decades. These long-lived plasma cells are the immune system's most devoted keepers, cells that have settled into the quietest corner of your body and dedicated their entire existence to the production of a single antibody, year after year, without interruption. Studies have shown that antibodies against smallpox can be detected in the blood of people vaccinated 50 or more years earlier, and similar longevity has been observed for antibodies against measles and other diseases. It means that a single encounter with an antigen, a single vaccination, a single infection, can leave behind a protective legacy that lasts a lifetime, a molecular echo of a moment that happened decades ago, still sounding in the blood, still offering its protection long after the original event has faded from conscious memory. The antibodies in a mother's breast milk are not fixed or static. They change over time, adapting to the infections the mother encounters in her environment. When a nursing mother is exposed to a new pathogen, perhaps through contact with a sick family member, or through the microbes present in the food she eats and the air she breathes, her immune system responds by producing antibodies specific to that pathogen, and within hours, those antibodies can appear in her breast milk. This means that the protection a baby receives through breastfeeding is not just a general immune boost, it is a dynamic, responsive, constantly updated defense that reflects the specific threats present in the mother and baby's shared world. If the mother catches a cold, her breast milk soon contains antibodies against that cold virus. If she is exposed to a new bacterium, antibodies against that bacterium appear in her milk. It is a living, breathing immune connection between two people, a system so sensitive and so responsive that it adjusts itself to the world in real time, offering the baby protection that is as current and specific as the mother's own immune response. By studying the DNA of living species like sharks, scientists have traced the origins of the antibody-based immune system back over 450 million years, not through fossils, but through molecular clocks and the genetic code still carried by modern descendants. Antibodies are made of proteins, which are soft molecules that degrade far too quickly to survive in the fossil record. So instead of looking for ancient antibodies themselves, scientists study the genes that encode them in the species that are alive today, comparing the sequences across distantly related animals and using mathematical models to estimate when those genes first appeared. The results point to the emergence of the adaptive immune system, including VDJ recombination and the first antibody-like molecules in the common ancestor of all jawed vertebrates, somewhere in the Ordovician or Silurian periods of Earth's history. It is a form of molecular archaeology, a way of reading the past through the living present, and it tells us that the system protecting you tonight is among the oldest and most conserved innovations in the entire history of animal life. Your antibodies are ancient, older than bones, older than teeth, older than the first forests that covered the land. The total number of antibody molecules in your body at any given moment is staggering, estimated to be on the order of 10 to the 13th power, or roughly 10 trillion individual molecules circulating through your blood and tissues and mucosal surfaces. If you could somehow gather all of these antibodies together and weigh them, they would amount to several grams of protein, a modest weight, easily held in the palm of your hand, and yet representing a collective intelligence that no computer has ever matched. And if you could somehow lay all of these antibody molecules end to end, stretching each tiny Y-shaped protein into a line, the total length would be almost unimaginably vast, a thread of molecular purpose winding its way far beyond any distance your mind could comfortably hold. This is the scale at which your immune system operates, trillions of individual actors, each one specific, each one purposeful, each one carrying out its task without direction or supervision, coordinated by nothing more than the ancient rules of molecular recognition and the quiet language of chemical signals. It is a civilization within you, invisible and immense, and it never stops working. Even while you sleep, especially while you sleep, your immune system is quietly producing and refining antibodies. Sleep is not a passive state for your body. It is a period of intense biological activity, a time when repair, consolidation, and renewal take place at every level, from the neurons of your brain to the cells of your immune system. During deep sleep, your body shifts its resources toward immune function, increasing the production of certain cytokines, signaling molecules that help regulate the immune response, and creating an environment that favors the activation of T cells and B cells and the production of antibodies. This is why sleep deprivation weakens the immune response and why restful sleep strengthens it. The night is not just a time of rest for your conscious mind, it is a time of quiet industry for your immune system, a shift change during which the body's most careful and important work is done. The antibodies being made inside you right now, as you listen to these words, are being crafted with the same care and precision that they always are, and the stillness of your body is helping them do their work more effectively than any waking moment could. And here, at the end of our journey, is perhaps the most tender fact of all. You have never been alone in your body. From the moment your immune system began to develop, before you were born, before you took your first breath, before you opened your eyes to the world, antibodies were there. They were there when your mother's IgG crossed the placenta and entered your blood, wrapping you in borrowed protection. They were there when your own B cells began their first tentative rearrangements in the bone marrow, assembling the genes that would give rise to your very first antibodies. They were there in the first sip of colostrum that coated your newborn throat, in the tears that washed your infant eyes, in every drop of saliva and every breath of air that carried IgA across your mucosal surfaces. And they are here now, tonight, in this moment, circulating through your blood as you rest, refining themselves in your lymph nodes as you breathe, standing watch at every surface where the inside of you meets the outside world. You have never once had to ask them to protect you. They have simply done it, quietly, faithfully from the very beginning, and they will go on doing it for as long as you are you. Good night. And so we come to the end of our gentle walk through the quiet architecture of your own protection. 100 small truths, each one a reminder that you are not alone in your body, that you never have been. Right now, as your breathing slows and the edges of the world grow soft, your antibodies are still there, patrolling, remembering, refining themselves in the dark. They do not need your attention. They do not need your worry. They only need you to rest, which is exactly what you are doing. So let them do their ancient, unhurried work. Let them hold you the way they always have, silently, faithfully, from the very first moment you were you. Good night.